Hi guys, welcome to this video. In this video, we're going to talk about probability in genetics. Right now, what is genetics? What is genetics? Genetics is actually a science that deals with how genes are transferred or inherited from parents by offsprings. And by so doing, you have discovered a new word genes. Genes, they are just units of heredity. Yeah. So they carry out the traits that are inherited from parents. So say the color of the skin, like the complexion, the color of the eye, the shape of the ear, and those kind of good traits are all transmitted by the genes. So genetics is all about studying how that transmission or that inheritance is done. Right. So um, over here we're going to talk about probability. Okay, the probability that a gene or a particular trait will be inherited by an offspring from its parents. So, um, what then is probability? To calculate probability is the likelihood of an event occurring, right? So, if the event occurs, then it's favorable outcome, right? So the favorable outcome divided by total outcome. So if, for instance, we have in a class we have 500 students, and then we have say 300 girls, then the probability of a girl being in that class or the probability of just finding a girl in that class would be the total number of girls, which is the favorable outcome, divided by the total outcome, which is 500 students. So that would be 3 over 5 for the student being a girl. How about a boy? If you have 300 girls and the total number of students is 500, then it means we have 200 boys. So the probability that you'll find a student who is a boy in the class will be 200 divided by 500. That will be 205. So this is a probability. And then how do we apply this in genetics? And what's the significance? Right? So usually in probability we have two main types of events. We have mutually exclusive events and then we have independent events. Right? So if you talk about mutually exclusive events, they are events that do not occur together, right? So when one is occurring, the other must might will not occur, right? So let's say let's assume rainfall, right? When rain is falling, we don't expect the sun to be shining. Though in some circumstances we have such a thing happening, but then the actual case is that when rain is falling, the sun will not be shining. So those kind of events, we call them mutually exclusive events. For example, tossing a coin. So a coin has two parts, that is the head and the tail. The upper part, calling the head, and the lower part, let's call it the tail. So when you toss a coin, it's either you get the head or you get the tail as the outcome, right? So if you want to, for instance, calculate the probability that you get a head, then so far as you have two outcomes, is either a head and or tail, then the probability of getting a head will just be 1 over 2. All right. And then the same applies for tail. Then what about independent events? What are they? All right. So mutually exclusive events, we said they do not occur together. All right. So the occurrence of one denies or stops the occurrence of the other. Right. So independent events, they are the opposite, where they can occur together at the same time simultaneously. So let's assume I have one coin in my left hand and another in my right hand. When I toss the two coins at the same time, what is happening in my left hand is independent or does not depend on what is happening in my right hand. Okay, so even if I get a head here or a tail, a head in the left hand or a tail in the right hand, those two things do not even depend on each other. What's happening here does not depend on what's happening here. So they can occur together. So I can toss the coins at the same time. We call those kind of outcomes independent outcomes or events. So that is that. Now, let's now bring that idea into genetics. Let's apply that in genetics. So the probability of inheriting an allele or gamete from parent is a mutually exclusive event. This is just an example of a mutually exclusive event. So, the genes have two forms. The forms of the gene, we call them the alleles. It's either a dominant one, they usually denoted by the capital K, and the recessive, which is usually denoted by the small letter. So, if you inherit, let's say for instance, you're talking about gene of tallness, or let's say gene of height, yeah, gene T, okay, gene T. Um, the dominant one will be capital T, and the recessive will be small t. So capital T means, since you're talking about height, capital T means you're gonna be tall, and then the small t means you're gonna be short. Right. So when you inherit the capital T allele from, for instance, your mother, you can't inherit the the small t from the same mom. All right. So it's either one. You're just gonna inherit just one of the gametes, the alleles from your mom. So then the other one from your daddy. So having your father. So that is how it happens. So those two events, so far as you can't inherit the two of them from the same parents, we call that mutually exclusive events or outcomes. So this is the illustration. So if T, capital T is for tallness and small t is for shortness, then the probability of you inheriting the allele or the allele for tallness is going to be 1 over 2 because the total outcome is 2. Right? It's either you are tall or short. So 2 outcomes. Then if you are tall, that means 1 over 2. The same applies for shortness. That will also be 1 over 2. Yeah. Alright, so monohybrid cross. Now let's go into crossing. See, I was earlier talking about mating, right? Parents inheriting, genes from parents, either mom and mother or father. Right. So, um, we can only inherit something, a gene or a trait from our parents only when they mate. Okay, they mate and then the gametes is formed. Right. The gametes are formed, and the zygote forms, then the zygote develops into the offspring. Right. So, let's assume now let's try to understand what a monohybrid cross is. A monohybrid cross is a cross between two heterozygotes, right? Uh, that are different, okay, at one gene locus. So one gene locus means you're looking at only one gene, okay, at one particular locus, right? So if you're looking at gene T, then the two forms of that gene are going to be the dominant capital T and the small t, that is the recessive. So monohybrid is gonna be capital T, small t, right? It means it's different at just one gene locus. It's just different at just one gene locus. So capital T, small t, if you take a cross between those two heterozygotes, we call it a monohybrid cross. And so making a cross between the two, you could have something like this. The genotype, the capital T, small t, will segregate into forming the alleles. Remember the law of segregation from Mendelian inheritance. So you're gonna have capital T, small t for one parent. That is the one in the blue line, the top. Then we also have capital T, small t for the other parent. So you have mother and father, right? So capital T will form, capital T. Then capital T, small t will form capital T, small t. Capital T and then the small t also form another capital T, small t. And then the small t combines to form small t, small t. So in one of my videos, we actually spoke about crossing over, recombination, actually another linkage. I spoke more on that. So this is recombination. After this celebration, the recombination occurs. And these are the possible outcomes. So over here, we are going to have 4 progenies, right? 4 offsprings, and each of them is going to have different different, how do you call it, different gametes or genotypes. Yeah, they're gonna have different genotypes. So capital T, capital T, capital T, small t, capital T, small t, small t, small t. So we can calculate the probability of an offspring having being formed from this kind of mating and having a particular genotype. Say what will be the probability of an offspring getting a genotype of capital T, capital T. So capital T, the total outcome, that is total population over here is 4. So 4, right? So to get that of the capital T, we count the number of offspring that have capital T as their genotype. And over here is only 1. So that becomes 1 over 4 for the probability of capital T. Then probability of capital T small t will be 2 over 4 because we have 2 of the progenies having capital T small t as their genotype. So that will be 2 over 4. So that will simplify to 1 over 2. The same applies for probability of small t. Only 1, okay, so 1 over 4. Alright, so maybe you all know of dominance. If you have capital T from one of the Mendelian inheritance, I mean, he spoke about dominance, right? Where if there's existence of the dominant allele, the small, that is the recessive, doesn't show its effect. So if you have capital T, and for instance, let's say T is for tallness and a small t is for shortness. So capital T is gonna be tall, right? Capital T, the offspring gonna have are gonna be tall in height. And if you have capital T, small t, because the capital T is having it can oppress or suppress in the effect of the recessive allele small t, the offspring is gonna be tall in height. Okay, that is dominance, complete dominance. Then we have the small t, because small t is for shortness, then the person is for short. Right. So let's move on. So number of different genotypes from this table, number of different genotypes. Since capital T is tall, we are going to have phenotype, that is the physical appearance. How does a person look like physically? So here, capital T, person going to be tall. Capital T, small t is also going to be tall. Capital T, small t is also tall. Then the small t is going to be short. So we have 3 of them being tall and then 1 being short. So the 2 different types of phenotype that we have here is 2. Right, the different phenotypes, the number of different phenotypes varies too. And then phenotypic ratio. Now, the phenotypic ratio compares the different phenotypes and then their ratio, their amounts. Okay, so from the table, we realize that 3 people are gonna be tall and 1 is gonna be short. So the phenotypic ratio becomes 3:1. All right, tall is to short. So that is it. Capital T, small t are all gonna be tall. Then the small t is gonna be short. So that gives us 3:1 as the phenotypic ratio. Now the number of different genotypes. That is, if you look at the table, the number of different genotypes are actually 3. So we have capital T, which is 1, capital T small t, which is another. Capital T small t is the same as the previous one, capital T small t. So we are going to take only one of them. So, and then we have small t. So we are going to have 3 different genotypes in this table. So number of genotypes becomes 3. Number of different genotypes becomes 3. Then what about the genotypic ratio? So if they are 3, then we have capital T is 2, capital T small t is 2, small t. Now we have 1 of the offspring having capital T. So we have 1 for the capital T. Capital T small t, we have 2 of them. And then small t, we have 1. So the genotypic ratio becomes 1:2:1. Right? Then what about the total population? Total population is the same as the total number of progenies or offspring that were formed. So from the table, we have 4 people, 4 offsprings formed. So that is that. Then let's move on to dihybrid cross. So in monohybrid, we could realize that we are talking about only one gene, right? Only one gene, where the offsprings or the parents differ or heterozygous at only one gene locus. However, here with the dihybrid, it's actually one that is different at two gene loci, right? We have two different genes where it's showing differences, okay, at both loci. So assuming the two genes are gene T for height and gene B for skin color. So this how them. So if it's heterozygous for height, it's gonna be capital T small t, and if it's heterozygous for skin, it's gonna be capital B small b. So the genotype becomes capital T small t, capital B small b for one parent. And since they are all heterozygous, or they are all dihybrid, then that becomes capital T small t, capital B small b, crossing capital T small t, capital B small b. Now, phenotypic ratio. Now, if you do the same thing that was done for the monohybrid, you could realize that the phenotypic ratio in this case is going to be 9:3 is to 3 is to 1. Right? What does it mean? The first 9 has some implication. It has a meaning. The 9 means that the first 9 represents dominance. Okay, two dominant genes. It means those offsprings have both the dominant genes, that is the T and the capital B, capital T and capital B in their genotype. So if you have something like capital T, capital T, capital B, that is one for the that is gonna count within that 9. So the 9 has at least one of the at least two, alright. It has both genes being dominant. So it has the dominant alleles for both genes. So for instance, even the parents themselves is gonna be considered as part of the 9 because in the parents where we have the capital T small t, capital B small b. For the T we have capital there, and for the B also we have capital. So for the 9 it represents two dominant genes, and the 3 represents one dominant gene. So if total number of different genes are 2 and one is dominant, it means the other one is also the other one is recessive. If one is dominant, it means the other one will be recessive. But then if both are dominant, it means we are having both dominants, right? Then the second trait also represents. So the first trait could be that the T is dominant and the B is recessive. And then the second trait will mean that the T is recessive and the B is dominant. And then the one represents recessive for both genes. So small t, small t, small b, small b. That is for the one. So both are recessive. So how to calculate the population? Now the population for the dihybrid is gonna be 16. 16. Now I'll shortly show you how to calculate these things. Now the number of different genotypes. How do you calculate or determine the number of different genotypes that are gonna form from a dihybrid cross? You use the formula 3 to the power n, right? Where the n represents the number of genes. Okay, so since we are talking about dihybrid, the number of genes here is gonna be 2. That is the n is gonna be 2. So you're gonna have 3 to the power 2, which will be 9. So for the hybrid, we have 9 different genes, having 9 different genotypes. Then we use the formula 2 to the power n to calculate for different number of different phenotypes. Okay, so if in the hybrid the n is equal to 2, then 2 to the power 2, that will be equal to 4. Right? That will be a number of different phenotypes. So at the phenotypic ratio, you can see that every number represents a particular phenotype. Right? So the first one represents the fact that the person will be tall, and if for instance the gene B, we are talking about capital B being black and small b being white, then the 9, all the members in 9 are gonna be tall and black. Then the first 3, if we are considering the T to be dominant and the B to be recessive, then for the T being dominant, the person is going to be tall and white. And the second trait, we are considering the fact that the T will be recessive and the B will be dominant. So over there, If the T is recessive, it means the person is gonna be short but black. Then the last one, the person is showing recessiveness in both genes. So the person is gonna be short and white. So that is that. Then the number of different phenotypes was calculated as 2 to the power n. So 2 to the power 2 is gonna be 4. Then number of progenies, that is the population, 4 to the power n. So 4 to the power 2 will give us 16 for dihybrid. We could use the same formulas for the monohybrid. We could see that to give us the same things as we got earlier. Alright, so we have a sample question. Now, the question says, in dihybrid cross, what will be the probability that an offspring will have genotype of capital A, capital B, small b. So, for instance, looking at this genotype, capital A means that the person is having a dominant A, and capital B, small b also means that the person is having at least one dominant B. So, this is the case of two dominant genes, right? So, this particular genotype will fall under the ratio of 9, as we looked at earlier in the phenotypic ratio. So, you have to calculate that. Then the second one, let's calculate the offspring, the probability that the offspring will have capital A, capital B genotype. Then it follows. So let's proceed to the solution. So before you could solve this problem, you have to identify the fact that the hybrid cross, since we are looking at gene A and gene B, the dihybrid cross is gonna be between capital A small a, capital B small b. That is gonna be one dihybrid. So I mean one yeah, dihybrid. So the cross is gonna be capital A small b, capital B small b, as against capital A small b, capital B small b. So that is gonna be the cross, the dihybrid cross. So to do this, you're gonna take the identical genes. So in the first parent, that is the capital A small b, capital A small a, capital B small b, and then in the second parent, which is also capital A small a, capital B small b, we're gonna take the identical genes. So gene A at both sides. At the first side, the gene A is having genotype of capital A small a, and then the second one too is capital A small a. So you cross capital A small a as against capital A small a. So that's what we've done in the table. Capital A small a as against capital A small a. So you end up getting these possible outcomes or genotypes for the progenies. Then you do the same thing for the B. So then you calculate the individual probabilities for all these genotypes. So probability for capital A from the table, capital A, we only have one of them out of the total 4 outcomes. So that becomes 1 over 4. Then it continues as we did for monohybrid. And it's clear that everyone will be able to do this. Now, to calculate for probability that the offspring gonna have capital A, capital B, small b, we consider the fact that what is happening at gene A, when there's segregation at gene A, is independent of what is happening at gene B. So the segregation at gene A is independent of the segregation at gene B. So since they are independent outcomes, we could multiply the probability of both genotypes, right, to get the probability of the dihybrid genotype. That is the capital B, small b. So we calculate for capital A. Earlier we had 1 over 4. Then the capital B, small b, we had 1 over 2, right? So since there are two independent outcomes, we multiply them. Okay, for independence, we multiply. For mutual exclusive, okay, so it's gonna be capital A, and capital B, small b. And whenever we have and, we multiply. So that is how it's gonna look like. Then the second one, capital A, capital B. Right. So over here, so you have capital A to be 1 over 4, capital B also to be 1 over 4. These again are independent events, so you multiply. You're gonna have 1 over 16. Then we apply the same principle here. Then we are to solve for the probability that you have the offspring gonna have two dominant genes. Okay, probability that the offspring gonna have two dominant genes. Now For two dominant gene, it means that we are at what? The 9. Right? I told you earlier that 9 represents two dominant genes. The 3 represents one dominant gene. So both traits represent one dominant gene. It could be the T or the B. Right? So the first one could be the T or the B, and the second one could also be the B or the T. Then the 1 represents no dominant gene. In other words, all recessive. So both genes are recessive. So that is two recessive. Right. So probability that the offspring gonna have two dominant genes, we just take the ratio for that, the two dominant gene, and divide by total uh outcome. That is the system. That is the progeny, right? 9 over 16 for that. That is clear. And then probability that the person gonna have one recessive gene. If you have one recessive gene, total number of genes here is 2. So if one is recessive, it means the other one is dominant, right? So one recessive, one dominant. That is the meaning. So one recessive, one dominant. I told you earlier that the three represents one dominant, one recessive, right? So it's either the first three or the second three. So this is an example of mutually exclusive. It's either the offspring is at the third trait, is part of the first trait, the ratio of the first trait, or is part of the second trait. So if the person is part of the first trait, the probability is going to be 3 over 16. And then if the person, sorry for that, if the person is part of the second trait, the probability is going to be 3 over 16. So the probability of one recessive becomes 3 over 16 plus 3 over 16. So you're going to get that is 6 over 16. Then probability of all recessive genes, all recessive. So no dominant. And no dominant, I told you the 1 represents no dominant in the ratio. So the probability becomes 1 over 16. All right. So that is that. Yeah.